classes where the educator comes online and they teach everything online. And you can join these classes and get your doubts clarified. If you haven't understood anything, you can always come here and ask all your questions. If you want to learn more, if you want to know things, if you want to know little more than what is being taught in live classes, you always have recorded sessions, and these sessions are really helpful because these classes have all the content that you need. These are very crisp and concise and to the point video classes. And we also offer notes which are based on the recent CBME curriculum, and MCQs which will help you in your upcoming next examination. These MCQs will be of really good use for you. And we also offer personalized counseling sessions where you will be given a buddy. So they'll be like a friend and who will make a study plan to you, and they'll sit with you to make sure that you stick to the study plan, and they'll analyze your strengths and weaknesses, and they make sure that you stay on track, and they make sure that you comfortably pass all your exams. 3 different subscription models are available. We have basic, standard, and premium. In the basic sessions, you have pre-recorded classes, live class, live doubt clarification, one-on-one guidance, notes, and in the standard plan, you have pre-recorded live doubt clarification, quick revision examination modules as well. But in the premium is like all-in-one, class where you get everything. Live classes, recorded sessions, doubt clarification, personalized guidance, notes, and quick revision exam modules. And you can also use my code Dr. Sushma 20 for any additional discount on all your subscription. Okay, yes. So this is the tentative schedule that we have, and let's start with development of week 3. As I keep saying, guys, embryology basically is like it's like a story, right? Up until now, what we have studied. So we started with a 2-celled zygote, which went on to become a blastocyst. Sorry, first it went on to become a morula, which was a 16-celled stage. From 16-celled stage, it entered into the uterine cavity and it became the blastocyst, right? Now, the cells of the blastocyst rearranged themselves and they gave rise to a hypoblast and an epiblast. So this is the epiblast and this is the hypoblast. And finally, we had the formation of 2 cavities. One is the amniogenic cavity and second is the secondary yolk sac. Amniogenic cavity is lined by amniogenic cells, and the yolk sac, primary and the secondary yolk sac, is lined by hypoblast cells, which is also called as Huesner's membrane. And finally, towards the end of week 2, we have the formation of extraembryonic coelomic cavity, and there is one amniotic cavity, and there is one yolk sac, and we also have the formation of extraembryonic mesoderm as well, right? The yellow shaded portion is the formation of the extraembryonic mesoderm. And then after the formation of extraembryonic mesoderm, we also had, the formation of the chorion frondosome here, which goes on to become the umbilical cord. And towards the end of week 2, we have basically cytotrophoblast and sensitrophoblast. They've already formed tertiary villi, and we have a amniotic cavity, and we have a secondary yolk sac. Then we have what is this? Extraembryonic coelom, and the entire is filled with extraembryonic mesoderm. So this is how the entire embryo now looks like. Okay, this is how it looks like now. Now, from here, what happens is the story of today's class. So again, just to revise, we first had a 2-celled zygote, which underwent cleavage to form a form a morula. Very good. This morula went on to become a blastocyst. This blastocyst further gave rise to two different cells. One is the epiblast and the other is the hypoblast. And finally, after so many changes, so many events that happened, we have the formation of this particular structure. From here, we'll see what happens. Okay. The joy of learning is as indispensable in study as breathing is in running. That means if you're not enjoying what you're studying, guys, then it's the joy of learning is as important as breathing, as in running. That is how important to love and to understand what you are studying. And this is more so when it comes to embryology. If you do not understand, if you are not enjoying learning embryology, then you will not, then there's no point in reading also. Correct? Yes. So with that, let's look at a clinical case scenario. We have a 35-year-old pregnant woman presents to her obs gynae for a routine prenatal checkup at 10 weeks of gestation. What is the gestational age now? It is 10 weeks. How many weeks have we completed now? We have come till the end of second week. Okay. From third week, what exactly is happening is we are seeing, right? She reports occasional nausea but denies any vomiting. On USG, the obs observes a single fetus with an omphalocele. This is the buzzword, guys. So what is this omphalocele? It's a condition where abdominal organs protrude through the defect in the umbilical cord. There is no evidence of any other congenital anomalies. What is the most likely cause of observed omphalocele, which we will answer when we come to the end of this class. So what is this gestational age given? 10 weeks. And what is omphalocele? Omphalocele is the abdominal organs protrude through the defect in the umbilical cord. But what to be noted over here is, in omphalocele, the omphalocele is actually covered by the membranes, whereas there is something called as gastroschisis, where the abdominal contents are exposed and they are not covered by the any membrane. Okay. Yes. Now let us just look at what is going to happen in this particular week of development, and then we'll come back to the previous question. All right. So this is kind of a summary that what we will be studying. So what exactly I am seeing in the first diagram? I'm seeing that there is an ectoderm. Okay. Already there's an ectoderm. Until now, what I was telling, there was an epiblast and there was a hypoblast, but now I'm having an ectoderm. Now I'm having something called as a notochord, but cavity is there. Amniotic cavity is still there. Then after here, again, I'm seeing some different paraxial mesoderm. And in the secondary yolk sac, there is mesoderm, and there are some cavities also present, present in the lateral plate. Then there is something called neural groove. Then we have the formation of a somite. So what are all these structures, guys? How my zygote from this structure that I had drawn in the previous slide, if you see, and there is extra embryonic coelom and everything, which again went bigger and bigger and made the amniotic cavity smaller, which we had studied in the previous class. From this, how has it become this? From this, how has it become this? From B to C, C to D is what we are going to be learning in today's class. All right. Now, the process of this becoming this is what is called as gastrulation. So what is gastrulation? Gastrulation is a process in which we have the formation of embryonic layers. Okay. Now, gastrulation begins with the formation of the primitive streak on the surface of the epiblast. So what happens, guys? I've already told you, I have something called as epiblast in the amniogenic cavity, and below that I have the hypoblast, right? And this is the secondary yolk sac. Now what happens is, some cells in the hypoblast, they start to get elongated like this. Okay, what is this tissue that is present below? This is the hypoblast. And what is this cell layer that is present above? This is the epiblast. Some cells in the hypoblast, they elongate, they form elongations like this. Once these elongations are formed, the cells which are present in the epiblast, they start running towards these elongations. The cells in the epiblast, they start running towards this hypoblast, and the cells which are running towards the hypoblast are called as primitive streak. They are called as the primitive streak. But will they run completely towards the end of the epiblast? No. They stop at a certain point. The point at which these cells stop is called as primitive node. The point at which the primitive stick stops is called as primitive node. Just to come back, how was the embryo looking like? It was looking like it had two cavities. Cavity number one, which is the amniotic cavity. Cavity number two, which is the secondary yolk sac. In the secondary yolk sac, I had, what are these cells? These are hypoblast, and these cells in the amniotic cavity are epiblasts. As we approach the further areas of development, the cells in the hypoblast undergo these elongations and forms the cranial end. This elongations is what exactly is forming the cranial end. Now the cranial end of the embryo is determined. Okay, and then cells in the epiblast, once the cells in the hypoblast are elongated like this, they start running towards this cranial end, and the cells who are running towards the cranial end are called as primitive streak. They do not run all the way till the cranial end, but they stop at a certain point, which is called as primitive node, which is called as the primitive node. Okay, now which process involves migration of cells through the primitive streak to establish the three germ layers? Gastrulation. Gastrulation is the process which helps to establish the three germ layers. All right, so this is exactly what I was talking about. So now look at it, guys. Basically, what the diagram that I'm drawing is like this, but the diagram that is shown here is, if I turn the diagram this way, this is how it looks like. This is how it looks like. And the upper portion is where the primitive streak is present. And then below you have the hypoblast. Okay. Now the primitive streak consists of, there is a primitive groove, there is a primitive node, and there is a primitive pit. This is the primitive pit. This is the primitive node. And sorry, this is the primitive pit. This is the primitive groove, and this is the primitive node. Correct? Cranial end is formed by how, guys? Just now I told you, cranial nerve end is formed when wherever the elongations of the cells in the hypoblast takes place, that end becomes the cranial end of the embryo. Okay. After the primitive streak formation, what does this embryo have now? It has a cranial end, isn't it? Now it has a dorsal and ventral surfaces, and also right and left sides. Guys, just for your understanding, I've kept it right and left sides, but actually the right and left sides is determined by the notochord. All right. At this point, what I'm explaining, at this point, the right and left side is not determined, but once the formation of notochord happens, the right and left is also determined. But for now, remember, cells in the hypoblast undergo elongations to form the cranial end of the embryo, and the cells in the epiblast start running towards this elongations, and that forms the primitive streak. Primitive streak has these three different, it consists of these three different things: primitive pit, node, and primitive groove. Okay, this is a primitive streak, and where it stops, it does not end all the way. This is the cranial end of the embryo, right? But it does not go up all the way till the cranial end, but it stops at some point, which is called as primitive node. All right, so look at it, guys. Look at it. This is where we have taken a section, and this is how it looks like. Can you see the cells in the hypoblast? which have elongated to form. Here you can see cells in the hypoblast, they'll elongate like this to form the cranial end, and the cells in the epiblast, they start running towards the cranial end to form the primitive streak, okay, and which is covered by extraembryonic mesoderm. Up until now, it is only covered by the extraembryonic mesoderm. All right. Yes. Now, let's look at the mechanism of gastrulation as such. Now, until now, whatever has happened, we had these cells and we had the formation of a primitive streak. And this is the cranial end of the embryo. And this is the caudal end. And now we have extraembryonic mesoderm outside. Now, what happens is, as these cells start running towards the primitive streak, they start going downwards like this towards the hypoblast cells, and it is called as, this movement is called as invagination. The cells in the primitive streak, the cells which are present over here, they start going inside. They start going inside towards this hypoblast cells, and this process is called as invagination. Once enough number of cells have invaginated, what they do is they completely displace the hypoblast, and that forms the definitive endoderm. Definitive embryonic endoderm is formed now. Initially, what did I have? I had a hypoblast and epiblast. Now, hypoblast cells elongated at a point which formed the cranial end of the embryo. Now, what happened? The primitive streak, cells in the primitive streak started running towards it, and that is a primitive streak, and it stops at a certain point which is called as the primitive knot. And these cells from the primitive streak, they start going downwards, which is called as invagination, and they completely displace the hypoblast cells, and that forms definitive endoderm. After the formation of definitive endoderm, more and more cells come, and then they completely form another layer over the endoderm, which is called as mesoderm. And then the cells of the epiblast are also completely replaced by the cells of the primitive streak to form the ectoderm. So what has happened, these cells start going downwards. These cells start going downwards. And let's say, for example, if I have to show, if I'll show in different color, guys. This was the epiblast, and which formed the primitive streak here. Primitive streak started going and replacing the hypoblast. So once they replace the hypoblast, and they start forming the layers of ectoderm, endoderm, and mesoderm. Once the hypoblast cells are completely replaced, it forms the definitive endoderm. On top of the definitive endoderm, they further invaginate, further divide, and they form definitive mesoderm. And on top of this, what do we have? We have the epiblast cells, which are also replaced by them to form the ectoderm. This is how the primitive germ layers are formed. This is how the germ layers are formed, guys. They are formed by primitive streak. They are formed by the primitive streak. Understanding? So the epiblast, through the process of gastrulation, forms all the 3 germ layers, and this is how it is formed. You can see the invagination happening, and the cells are migrating, and they are removing the cells of hypoblast and forming the 3 different layers. This is a much better picture. And this is extraembryonic mesoderm. Now I have the intraembryonic mesoderm, which is also called as the definitive mesoderm. Okay, definitive mesoderm. All of these are the permanent structures from which the derivatives now start to begin. All right. From this inner cell mass, amniotic cavity was formed, and all of the extraembryonic membranes and everything got formed. And now I have the formation of the primitive streak, which is forming definitive layers. Okay. Now, as the more and more cells move between epiblast and the hypoblast layers, they begin to spread laterally and cranially. And gradually, they try to migrate beyond the margin of the disc, and it finally establishes the contact with the extraembryonic mesoderm covering the yolk sac as well as the amnion. All right. In the cephalic direction, what happens is they pass on each side of the precaudal plate. But It does not cover the precaudal plate, guys. So as the the intraembryonic mesoderm, the mesoderm is as it's spreading laterally and as it's spreading downwards and upwards everywhere, it does not cover two regions. One is the precaudal plate, okay, which forms the oropharyngeal membrane, and second is the buccopharyngeal, sorry, oropharyngeal membrane of the cranial end of the disc. And then it does not form, it does not cover the cloacal membrane, which eventually goes on to form the anal cavity and the rectum and the anal cavity, whereas the buccopharyngeal membrane forms the mouth. As it spreads, it just does not cover these two areas, which are called as the oropharyngeal membrane and the cloacal membrane, but it covers everything else, and it even makes a contact with the extraembryonic mesoderm. Understanding? So as you can see, it is not covering the cloacal membrane and oropharyngeal membrane, and these two openings remain as such, and finally they form the mouth and the anal canal. Okay, this is how the primitive streak is giving rise to different layers, and these layers are surrounding everything except for two openings. How many openings we have in the GI canal? We have two openings, mouth and the anal canal. So it makes sense that the mesoderm is not spreading and it is covering the oropharyngeal membrane and the cloacal membrane, isn't it? Had the mesoderm spread laterally and cranially and it covered up oropharyngeal and cloacal membrane, then we wouldn't have had these two openings. It only logical and makes sense that the mesoderm, the developing mesoderm, will not cover oropharyngeal as well as the cloacal membrane. And it is very logical. That is why we have mouth and an anal canal. Okay. Which of the following structures is responsible for the formation of three germ layers during gastrulation? Primitive streak. This is a very, very important concept, guys. Just to recap, we had cells in the epiblasts and cells in the hypoblast. Hypoblast cells underwent this elongations. Then the epiblast cells started running towards them. They underwent some differentiation and they formed the primitive streak. Primitive streak started to invaginate and they replaced the hypoblast cells with definitive endoderm. Above endoderm, it accumulated to form the mesoderm. Above mesoderm, it completely replaced all the epiblast cells to form the ectoderm. Then this mesoderm is the definitive mesoderm. Okay, it starts spreading cranially, caudally, laterally, and makes a contact with the extraembryonic mesoderm as well. And it goes and reaches till the cranial end, and also laterally it starts spreading to meet the extraembryonic mesoderm. But it does not cover two openings, or it does not cover two places. One is the cloacal membrane, and another is the buccopharyngeal membrane. Okay, and this cloacal and the buccopharyngeal openings eventually go on to form the mouth and the anal canal. Okay. Sorry, oropharyngeal, not buccopharyngeal. Oropharyngeal, which is also called as buccopharyngeal membrane, by the way. Okay, you can see here how the buccopharyngeal membrane and the cloacal membrane are not covered by the mesoderm. You can see how the mesoderm is spreading laterally, spreading, it goes ventrally and it spreads laterally and mixes with the extraembryonic mesoderm as well, except for two places where buccopharyngeal and cloacal membrane. So this process is what is called as gastrulation. This process is called as gastrulation. Cells from the epiblast form primitive streak, invagination, formation of three layers, mesoderm starts spreading, right? It spreads everywhere, makes contact with extraembryonic mesoderm. It does not spread in two areas. One is the buccopharyngeal membrane, another is cloacal membrane, which go on to become the mouth and the anal canal. It only makes sense. It's very logical. It spreads everywhere. That's why you have from the mesoderm, muscles, bones, and skin. It is present everywhere, except for in these two areas. That is where we have the opening, because it is not covered by muscle. It is not completely sealed off by muscle, and then what else? Bones. So that is why it does not go and cover the buccopharyngeal membrane and cloacal membrane for the formation of mouth and anus. Okay. Yes. Now, gastrulation itself may be disrupted by so many genetic abnormalities. One of the things is caudal dysgenesis or cyanomelia. What happens is, suppose if the mesoderm does not spread everywhere else, it does not, the entire process, if the invagination is not taking place, correct? Why this happens is because there is mesoderm which is insufficient in the caudalmost region of the embryo. In the caudalmost region, what did we have? We had this cloacal membrane in the caudalmost region, where it is not possible to have, where the the mesoderm is insufficient. It results in this condition known as, this is called as cyanomelia. Cyanomelia, where there is no development of the limbs. But as you can see, above this, the baby looks normal. Look at the limbs, look at the head. Of course, you can see some deformities, but overall, grossly speaking, this looks normal. Okay. Yes. Now let's look at the formation of the notochord. Now, notochord is one structure, guys, that determines not only the axis around which the embryo develops, but also right side and the left side. Okay. Now, if I have to show where exactly notochord is, I told you that we have the formation of ectoderm, mesoderm, and endoderm. Notochord is formed between the ectoderm and mesoderm. Right. It is forming on top of the mesoderm. So obviously it has to be derived from the mesoderm only. Correct or not? Now, why is this notochord formed? It is the basis of the axial skeleton. This actually notochord is the axis of the embryo. It not only dictates, right side and left side, but also it also forms the axis, that is, which is cranial end, which is caudal end, around which axis the entire structures are developing. So basically, there are these steps involved in the formation of the notochord. We have the precaudal plate formation, the notochordal process, notochordal canal, neuroenteric canal, notochordal plate, and finally, definitive notochord is formed. How all of this takes place in itself is a very big topic which I cannot cover in just one class, but just know that, guys, where exactly the notochord is formed. This is ectoderm. Let's say this is mesoderm. This is endoderm. It is formed here between ectoderm and mesoderm. Which layer gives rise to notochord? Mesoderm gives rise to notochord through all of these processes. Okay. Next, notochord serves as the basis for the axial skeleton, which I've already discussed, and process of the notochord starts by the appearance of the precaudal plate. All right. Now, the precaudal plate forms between the tip of the notochord and the oropharyngeal membrane. What is the significance of oropharyngeal membrane, guys? It is not covered by the mesoderm, and it lies caudal to the oropharyngeal membrane, derived from some sort of first cells that migrate through the primitive node in the midline and move in the cephalic direction. Okay, so basically, if one diagram is to know where exactly notochord is present, is this. The other diagram is, this is the precaudal process. Here is the primitive streak which is running like this. Here is the primitive streak which is running like this. Let me just take a different color for this. This is the primitive streak which is running like this. And then the most caudal is the precaudal plate. And then here is the, here we have the oropharyngeal membrane. Then notochord is present over here. Notochord is present over here. So this is where the notochord is present. This is the notochord. Same thing, if you rotate it like this is how it looks. In this diagram, whatever this diagram I have written, right? That is how it looks if I rotate. All right. Yes. Now look at this. Look at this diagram, guys. So just a second. Look at this diagram. So this is the primitive pit, primitive node and everything. And finally, towards day 18 and 19, we have the formation of notochord here. Can you see? Can you see? Here, this is a better picture, I can say, right? We have the primitive streak, primitive pit, and this is the precaudal plate. This is the neural plate and notochordal process. Can you see here? Can you see here? And this is a definitive formation of the notochord. All right. Now, notochordal process extends cranially to the precaudal plate, caudally till the primitive pit. So, if I have to write If you have to write this in an order, so that is how it looks like. This diagram. Okay, this is the precaudal plate, oropharyngeal membrane, notochord, and primitive pit. This is a primitive, not the cranial end of the primitive streak. Okay, next. The remaining roof forming the notochordal plate, which proliferate and detach from the endoderm and then form a solid cord of cells called as definitive notochord. All right, which underlies the neural tube. Now, neural tube, how it is formed, the elongation and the folding of the neural tube will be taught to you in the next class. But for now, what you must know is notochord is formed between ectoderm and mesoderm. Here is the notochord. In this section, if this is the cut section you are looking at, but if you turn it and look at it, if you remove the ectoderm and then you look at it, how does it look? Here you have the precaudal plate, oropharyngeal membrane, notochord, primitive streak. Two sections in which you can actually see this. What are the two sections in which you can see? One section is like this, between the ectoderm and the mesoderm. Other section, just remove the ectoderm and see from above like this. Then you will see the precaudal plate, then the oropharyngeal membrane, and then the notochord, and then you see the primitive streak over here. Two sections in which you can study notochord. All right. The formation of the notochord is crucial for what? Establishing the right to left symmetry and also providing structural support and signaling cues, because notochord is the one which stimulates the ectoderm to form the neuroectoderm, but more importantly is to determining the right to left symmetry. Next, let's just have a quick discussion on the allantois as well. So basically the allantoenteric diverticulum is a small diverticulum that extends from the posterior wall of the yolk sac to the connecting stalk. Okay. Now, this diverticulum or allantois appears around 16th day of development when the cloacal membrane appears. All right. Now, what is cloacal membrane? Cloacal membrane is formed at the caudal end of the embryonic disc. Is it or is it not? Cloacal membrane is also called as what? Sorry, cloacal membrane is actually formed at the caudal end of the embryonic disc, and this membrane, which is similar to structure of the oropharyngeal membrane, which is present in the cranial end of the embryo and does not have the intervening mesoderm. All right, understanding what is this diverticulum that extends from the posterior wall of yolk sac to the connecting sac, right? And the diverticulum appears when the cloacal membrane appears. Humans, it is rudimentary, but may be involved in the abnormalities of the bladder development. All right, this is where the allantois is. I know these diagrams look very difficult and you will not be able to orient to it, but at least you should know where amniotic cavity and amnion is present. So on the upper in the lower side, you will see the yolk sac. That much at least you should have an orientation to. All right. Otherwise, it becomes very, very difficult as we go study. Next, the growth of the embryonic disc. Now, initially, the embryonic disc is flat and it's almost round, and gradually it becomes elongated with broad cephalic and a narrow caudal end. All right. Now, the expansion occurs mainly in the cephalic region. That is the region of the primitive streak remains more or less the same. Okay. Next, the growth and elongation of the cephalic part of the disc are caused by continuous migration of cells from the primitive streak region. Is it not? Primitive streak, these are the cells that are running towards the caudal end, cranial end. Is it or is it not? Where is the primitive streak formed? It is formed at the caudal end. Primitive streak cells, they run towards the cranial end because the elongation of the hypoblast was formed, which determined the cranial end in the first place. So obviously cranial end is the one which is getting so many cells, so many number of cells which are coming towards the cranial end, whereas the caudal end remain as it is, more or less. Okay. Then primitive streak invagination also from surface cells, it goes downwards and migrates downwards and forms all three different layers, and subsequently the migration forward and laterally continues in the form of what? Mesoderm. And at this stage the primitive streak actually disappears. Once this complete Spread of the mesoderm is over. Okay. Next, in the cephalic part, the germ layers begin their specific differentiation by the middle of the third week. In the caudal part, differentiation begins by the end of the fourth week. Makes sense, right? In the cranial end, you have so many different layers, so many cells coming in, whereas caudal, less number of cells, so it begins a little later also. Right? Thus, gastrulation or formation of the germ layers continues in the caudal segment where cranial segment structures are still differentiating. Understanding gastrulation as a process of the formation of the membranes itself is still continuing in the caudal segments for one more week, whereas the cranial end already starts to differentiate. Correct? Why? Cranial end has more number of cells. More number of cells are running towards the cranial end. So the differentiation is also happening faster there. But here, still the accumulation or still the formation of the membranes is taking place. Okay. This is how the embryonic disc growth takes place, where you can see towards the end of the 20 days, the cranial end is far bigger as compared to the caudal end. You can see the primitive streak here and the notochord formation, and here is the cardiac area. All of this can be seen here. But what is to be understood is cranial end differentiation starts earlier than the caudal end. Point number one. Point number two, primitive streak completely disappears once the spread of the mesoderm is over. The job of the primitive streak is over. Understanding? Understanding? Yes. Let's take a small question. The process of gastrulation is characterized by formation of blastocele. Of course not blastocele. It got formed when? Towards the end of 6th or 7th day itself. Migration of cells to form neural tube. Let's see, question mark, looks like it. Conversion of bilaminar disc to trilaminar disc. Is it or is it not? In the gastrulation, what exactly happened? Invagination took place from two layers, from epiblast layer and hypoblast cells. We now have three different layers. We have ectoderm, mesoderm, and endoderm. So gastrulation is a process in which the bilaminar disc is converted into a trilaminar disc. This is the right answer. Formation of yolk sac. No, yolk sac got formed then itself. Then only the yolk sac, primary, secondary yolk sac were formed. When? Second week of development. Okay. Just to summarize today's class, guys, just to give a complete summary of whatever we had learned. So initially, in this first beginning of the third week of development, we had two layers. First layer is the epiblast, and below that we have hypoblast. This is the amniotic cavity. This is the yolk sac. Okay. Then, and of course, in the outside we have the formation of syncytial, cytotrophoblast, tertiary villi formation, and all of that is over. Come to the inside of the cell to these two layers, and some cells in the hypoblast area, they undergo these elongations to form the cranial end of the embryo. Once this cranial end is formed, the cells in the epiblast area, they undergo differentiation and they start running towards the cranial end, and this forms the primitive streak. And this forms the primitive streak. Okay, as it starts running towards the cranial area, it results in the formation of the primitive streak. Understanding what happened? Here I have the hypoblast cells which underwent these elongations, and the cells started running towards them. Those are the primitive streak. Then what happened? These are epiblast cells and these are hypoblast cells, and primitive streak is formed like this. The primitive streak which is formed started invaginating and it started going downwards. It's the primitive streak which is formed. As it starts invaginating, started going downwards, started replacing the hypoblast. Then they formed mesoderm. This completely replaced the epiblast also. And finally, we had the formation of a trilaminar germ disc, which is the first layer is ectoderm, second layer is mesoderm, third layer is endoderm. And also, what have we determined? Cranial end and caudal end. Then the mesoderm that is formed, I'm just continuing it over here, started spreading everywhere. If suppose this is the ectoderm, this is the mesoderm, and this is the endoderm. Let me just shade the mesoderm. Let me just shade the mesoderm. Okay. Now the mesoderm that is formed over here, it started spreading everywhere. It started spreading, it started going caudally, started going cranially, it started going laterally, and it also met with the extra embryonic mesoderm as well. But as it started spreading and covering every bit of the embryo, at two places, it stopped and it did not cover them. What are those two places? One is the buccopharyngeal area and another is the cloacal area. And these two places where the mesoderm did not exist went on to form the mouth and the anal canal. From then, we had the formation of the notochord between the ectoderm and endoderm. So this is where the notochord formation took place by the mesoderm. And you have to show where exactly it is present: precaudal plate, oropharyngeal membrane, and this is where the notochord is present. And here is the primitive streak, and this is the cloacal membrane. Finally, how the embryonic disc started developing? More and more cells, they started running towards the cranial end. So the cranial differentiation started off towards the end of second week itself. But then the caudal end, and also it started growing laterally also. So the cranial end became bigger, but the caudal end is still in the process of differentiation and it is much smaller than the cranial end. And this is how the development has taken place up until now. Up until now. What is that we are discussing? Towards the end of third week. Okay, towards the end of third week, these are all the different things that have happened. From here, we will see in the next class how different layers are, different structures are formed from these layers: ectoderm, mesoderm, and endoderm. We will see what are the different structures formed. We'll study about somites and pharyngeal arches, somites, and everything in tomorrow's class. All right, guys. Thank you so much for watching. I hope you're finding these classes useful, and I really wish you guys all the very best. And do keep studying, guys. Do keep attending classes and go back and study what exactly was taught in the classes so that you have a continuation. You know, embryology is all about having continuation. All right. Thank you so much, guys. And I wish you guys all the very best. Keep studying. I'll see you in tomorrow's class. If you have any doubt, you can follow across all of the social media handles and ask us. All right. Thank you.